we have learned different structures in protists, different nutritions, and other traits that are important for you to remember, and some important processes that have led to the diversity in protists. And very importantly, the primary and secondary endosymbiosis. Now we are going to jump into the reclassification of protists. And remember, we have 4 supergroups: excavata, the SAR, Archaeoplastida, and Uniconta. Excavates are protists that have a modified mitochondria, and they are going to have also a very particular flagella. So if you have to remember something from this group, is that they have a spiral or crystalline rod inside flagella. And also some of them, most of them, have a feeding groove, and that's where the name excavata is coming from. So they are excavated, and that's in one side of the body. This is a monophyletic group. Excavata is monophyletic. That means that we have the ancestor, and all of the descendants that we call excavates are descendants of these, all of them. This group is further split in diplomonads, parabacills, and euglenozoans. Most of them, as I said, they have flagella, feeding groove, and some of them, diplomonads and parabacillids, are going to be amitochondriated. And that doesn't necessarily mean that they don't have mitochondria. In the past, we used to think that they lack mitochondria, and therefore we thought that they were some type early evolving eukaryotic cells. But that's not actually true. They actually have a reduced mitochondria that we were not understanding before. So this amitochondriated group now, it lives mainly, some of the diplomonads live in anaerobic environments. Remember that anaerobic means that they cannot tolerate oxygen. Aerobic likes to live in oxygenated conditions or in oxygen conditions. But anaerobic don't like to be living in oxygenated conditions. One example of these diplomonads is Giardia intestinalis. Giardia lambda is another one. But Giardia intestinalis can be very dangerous. We can acquire, it's a pathogen to humans, and we can eat it from things that are contaminated or from water that is contaminated with Giardia intestinalis. And it causes severe diarrhea, and it could be really severe. So the other thing to remember is that the mitochondria, these reduced mitochondria, in this case, is called mitosomes. In contrast, the parabacillids are the mitochondria, which is also reduced, is called hydrogenosomes. This is one example of these parabacillids. And this is the same as Trichomonas vaginalis. As you can imagine, Trichomonas vaginalis can affect the vagina in women. It also can affect the urinary tract of men, but it doesn't cause severe symptoms. Dr. Angelica Riestra is a recent professor, or is an assistant professor in the biology department, and she studies the molecular mechanisms of Trichomonas vaginalis, in particular the pathogenesis. So if you are interested in some of these topics, you should be contacting her. The third group of excavata is the euglenozoans, and this is further divided into kinetoplastids, and the other one is the euglenids. So these euglenids are the same, or is the same group that I was presenting before when I introduced Ernst Haeckel, and this is the one that can be mixotrophic. So that moves with an amoeboid. It has an amoeboid movement and also flagellated movement, but it could also, mixotrophs refers to their ability to photosynthesize, right? Because they have all of these chloroplasts in here, but also is able, in absence of light, for example, and these chloroplasts won't be working, and then it has to switch to be heterotroph and become predator. It's going to have an eye spot in here, and that is going to help with detecting the light, and it responds to this, to the light, and the response is going to be also affecting the behavior. So remember, I told you it has phytotaxis, which means that it's going to follow the light, and that it wants to follow the light because if it's exposed to the light, it's going to photosynthesize. In the kinetoplastids, they have a single large mitochondria, and a lot of them can be parasites. And one example is trypanosoma, which causes sleeping sickness. In this picture, we can see the kinetoplastids along some red blood cells in humans. These are examples of some parasitic kinetoplastids. They belong to the trypanosome or the Trypanosoma genus. One of them can cause the Chagas disease, and the other one is the one causing the sleeping sickness. In this case, the vector is the tsetse fly, and that's the Trypanosoma brucei. Kinetoplastids have a particular mitochondria that has organized mass of DNA, and the whole thing is called the kinetoplast. They can live in fresh water, in marine environments, or in moist terrestrial ecosystems, and they are free living. So it's a very important group, these kinetoplastids. Now we have covered the excavata, and now we're going to jump into the SAR. And SAR is just the abbreviation of 3 different groups. That's stramenopiles, alveolates, and rhizarians. So that's the SAR, where the SAR is coming from. This group is monophyletic, again, having an ancestor with all of the descendants in that group, in the SAR group. However, this one is the most controversial of the 4 supergroups. In this case, in this SAR group, we know for sure that there was an event of secondary endosymbiosis that happened at the base of these clades. So these secondary endosymbiosis occur by engulfing red algae. The clades that descend from this ancestor, which engulf the red algae, are stramenopiles, and alveolates. So let's learn first the stramenopiles, which have these 3 different groups. Stramenopiles actually means straw hair, and so pile is coming from this hair structure, and that refers to the flagella. They are going to be photosynthetic, although some are heterotrophs. And the ones that are photosynthetic is because what I explained, that they engulf this red algae, which already has a chloroplast in it. They have 2 flagella. They have heterocon cells, and they have 2 flagella. And some of these flagella can white plash, and the others tinsel, or just tinsel. And you can see one of them is smooth, the flagellum is smooth, and the other one is hairy. Examples of these are diatoms, brown algae, omycetes, and golden algae. So one of the very exciting groups is these diatoms. They are unicellular, and they are kind of cute because they have this box appearance. It's almost like you have a box of shoes, they're closing them. And this, the outside structure is made out of silicon dioxide. And this is one of the valves, and this is the other valve. Similarly here, we have one valve and another one in here. So this is glass-like, and this structure is really hard and also helps to fossilize. So diatoms often are indicators of some strata in the fossil record. Very interestingly, diatoms are a major component of the phytoplankton. So they are these microorganisms floating in fresh water or in the sea, and they are primary producers. So they are going to be feeding a lot of other, the zooplankton, and the zooplankton later is going to feed other larger species. So they are a very important element in this cycle, the nutrient cycle. And they have all of these shapes and more. So it's a very large group, very diverse. They can also be important for diatomaceous earth. As I mentioned, they fossilize very well, and some people harvest them or mine like a mineral. because of their properties, structural properties, and they often are used for filtration or to polish, right? They use these silice to polish. The other group is the golden algae. They have this appearance. They look yellowish, brownish, because of the presence of carotenoids, and these are pigments. They have two flagella often in one of the extremes and can be unicellular, or in this case, we have a colony of brown algae. This is Dinobrion, is the golden algae that lives in fresh water. The third group of the estremenophiles are brown algae, and they look similar like plants. They are actually, the structures are very, they have evolved by convergence. So let's look at this. This is the holdfast, and the holdfasts help these algae, this brown algae, to anchor to the floor in the sea. And we have this type, which could be equivalent to the stem, but this is not the stem, and these are the blades, which look like leaves, but they are not actually leaves. They are called blades. And I'm emphasizing that they look similar, but they are not. That's because these algae don't have tissues, and therefore they don't have organs. We're going to learn later that plants do have tissues and they have organs. So they have a very complex structures, whereas this is just a mass of the same type of cells that have evolved independently. So they look similar, but they are not the same compared to plants. Some of these brown algae also have bubbles in the blades or at the tips of the algae, because that helps with flotation. So that helps them keep afloat. And if they reach the surface in the sea, that will help them to photosynthesize. So that's an adaptive structure. If you want to study these creatures, you should contact Dr. Tinsdale. She's a specialist she studies microorganisms and seaweeds, and also Dr. Edwards. This group is going to have alternation of generations. And from now on, this is going to be very important for you to remember. So pay attention to this concept, because alternation of generations are the alternation of multicellular haploids, right? Multicellular haploid and diploid generations. We don't have that in humans, and sometimes it's really hard for us to understand these concepts. But I'm going to be guiding you through for you to understand them a little bit more this class and the following. Some species can be heteromorphic and others isomorphic. So when we have one generation that is multicellularly haploid, and then we have the next generation that is going to be diploid, and it's also multicellular, but they look exactly the same, that's what it's called isomorphic. Whereas if they look different, they are called heteromorphic. It's very simple. Let's look at one example. So this is the alternation of generations in Laminaria. This is an algae. This is a brown algae that, let's start here. So this sporophyte is going to divide, is going to produce the spores. So the spores are always going to be a result of meiosis. So once that these sporangia, some cells in this sporangia undergo meiosis, the result is these zoospores. The zoospores now are haploid. Meiosis is always going to produce these haploid cells. And we're going to have these zoospores. They have flagella right here. And this zoospore is going to divide through mitosis now, and it's going to form a multicellular structure. You see, it forms this female gametophyte and male gametophyte over here. And each of these gametophytes is going to form now the gametes. And these gametes are going to fuse. So here the female gametophyte forms the eggs, and the male gametophyte forms the sperm. So the sperm swings and fertilize this egg. The fertilization always is going to reestablish the ploidy. So now it's going to be two-end, so diploid. And you can see in pink, it's everything that covers the diploid cycle, or the cycle with diploid condition. And in green or bluish, we have everything that happens that is haploid. Okay. So once that fertilization occurs, as I was saying, now we have a zygote that is 2N, and that divides and produces this massive individual, and that's the brown algae that we're more familiar with. I want to ask you, this multicellular individual looks identical or similar to these multicellular haploid individuals? I hope that you say no, because they are different. So in that case, we are talking about a heteromorphic cycle, right? They are heteromorphic. If these structures were similar, the diploid and the haploid, the multicellular structures, we will have an isomorphic species. But this one, Laminaria, is actually heteromorphic. Another group of these SAR is the alveolates. Right, we are done with the straminopiles. Now we're switching into the alveolates, and we're going to see these three groups. So dinoflagellates, apicomplexans, and ciliates. Alveolates, all of these guys are going to be grouped by the presence of sacs. So they have a membrane-enclosed sacs that we call alveoli. And the alveoli or alveoli are right here. So this is the flagellum, and you can see some of these alveoli. That's where the name is coming from, the alveolates. So we have some of them, dinoflagellates, and other parasitic examples in apicomplexans that we're going to explain. And here we have some ciliates, like the stentor that I mentioned before, and others. And you can see how they move, and they now are filtering some of the nutrients or other tiny cells that they can eat. What is this? So this is the red tie. And red tie is caused by the presence of dinoflagellates. Dinoflagellates are algae. And these algae can cause severe effects in the ecosystems. When they have a bloom, when they bloom, they reproduce like crazy because there is more nutrients, and maybe the water gets a little bit warmer. They can reproduce massively, and they also produce a toxin. This toxin, when it's being eaten it's filtered by mollusks. The mollusks eat the dinoflagellates and acquire this toxin, and then it can be passed to the next level. So some larger animals that live in the ocean can eat these mollusks now, and they can get sick. But we can also get sick if we eat this contaminated fish with this toxin, and it can be very bad. So we avoid eating fish that has been exposed to these blooms. They form or they produce this neurotoxin. So it has it damaged the nervous system in humans. However, this is the same dinoflagellate that I mentioned at the beginning of the class that I explained that is bioluminescent. And these bioluminescent get expressed when there is a sudden move in the environment. So waves, or if you are swimming in the ocean, when there is a bloom, you can actually see these dinoflagellates moving around you. So it's a really nice feature, but even you can make the move if you go and well, you swim or if you surf in these waves. So it's very cool. We have had them in San Diego. We have this bloom in San Diego this year around April, and sometimes we see them around those times. So dinoflagellates possess the plastids with chlorophyll. They have a lot of carotenoids, and that's why they give this color. And when we have them in high abundance, we can actually see the color of the sea changing. And it's an important component of the plankton, as I was mentioning before. So they are primary producers. The second group is the apicomplexans. These group are going to have the tip of this cell. This is the apical complex. That's why they are called apicomplexans. The apical complex is used to perforate cells that they are infecting or affecting. So apicoplasms are parasites, and they have this organelle called the apicoplast, and that's an adaptation that this group has. And this apicoplast has been originated from the engulfment of a red algae. One of the examples that I want you to remember is Plasmodium. Plasmodium is an apicoplexan that causes malaria, and it requires an intermediate host, and these hosts are the mosquitoes. This mosquito is going to be biting us and transmitting this Plasmodium, which affects the liver, and then it goes into the red blood cells, and it exits. And when we get another bite from the mosquito, that mosquito can carry its now the gametocytes that we created in our own body. So it's a very tough disease. There is a lot of people, thousands of people who die every year. For spring break, you decide to go to Thailand. There you are bitten by a mosquito. You get paranoid and catch the mosquito and take it to the lab. You confirm the mosquito carries Plasmodium. However, you never develop the symptoms of malaria. What is the most likely explanation? A, you have the immune system of Superman. B, mosquitoes suck at transmitting malaria. C, you are taking streptomycin. D, you might have sickle cell anemia. E, nobody likes you, not even mosquitoes. So this is not a possible scenario. Okay, so hopefully you remember that the sickle cell anemia confers some protection against malaria. The other group that we are going to be looking into is ciliates. And these ciliates have cilia. So this is one of the movement or the structures involved in movement. And hopefully you can see in all of these structures, in all of these species, we have tiny hair-like structures called cilia. And as I mentioned, they can be used to move, but also to feed. These ciliates, on top of having the cilia, they are going to have also micronuclei and macronuclei. So this is quite bizarre. So they have several nuclei in their cells, and this macronuclei is obviously larger, but it's actually a micronuclei that later grows and grows, and it takes charge of all of the function of these cells. So it's quite interesting. This is one example, Paramecium. And they also have a contractile back wall that uses to excrete things that they don't need. And they are mainly predators, eating other protists or prokaryotes. So we have covered alveolates. And now we're going to move into the third group of SAR, that's rhizarians. And rhizarians many of them are amoebas. Amoebas is another artificial group. So amoebas was defined as something that have this amoeboid movement. But now we'll see that some of these amoebas fall into this group in their rhizarians. And we're going to learn that other amoebas fall into the uniconta. So it's not monophyletic, it's an artificial group. In the case of the rhizarian, the amoebas have a pseudopodia, but they are going to have these extensions of the surface that are thread-like pseudopodiads. So it's not like the other video that we saw, but it's more like a thin pseudopodia. In this group, we're going to have the forams as well. Forams are protists that have tests, and this is the structure of the test that is a porous shell that is made out of calcium carbonate. So it's also very important to date sedimentary rocks. Forams have that advantage. And another group is the cercozoans. The third group of rhizarians that I want to present today is cercozoans. This is Polynella chromatophora, and these species have a chromatophore. let's look into that a little bit more. So this species, it's going to have a sausage shape structure, and this is the one that is photosynthesizing. So it doesn't look like our regular plastids that we are more familiar with, but it's more like a sausage type plastid. So that's called the chromatophore. And one very important or interesting feature of the chromatophore is that they have a layer of peptidoglycan. Does that sound familiar? What other things have peptidoglycans? So bacteria have peptidoglycans, and in particular, we have talked that the gram-negative bacteria, such as cyanobacteria, will have the peptidoglycan in a sandwich approach in between two membranes. Okay, the chromatophore has this peptidoglycan layer, and that suggests that they are derived from a bacterium. Now, this is probably the only example that we have that has evolved from primary endosymbiosis, different of the primary endosymbiosis that led to the evolution of all of the plastids in algae and in plants. So it's another event of primary endosymbiosis. That's what we think of Paulinella chromatophora. Now, the Archaeoplastida is the third group that we're going to cover. And Archaeoplastida is made out of red algae, green algae, and plants. And the ancestor had the primary endosymbiosis. And then this primary endosymbiosis led to the evolution of red algae and green algae. So all of the members in Archaeoplastida, the archaeo, ancient, and plastida, the plastid, that's the group that has the ancient plastid. And that's the one that has the primary endosymbiosis. So they are photosynthetic. Archaeoplastida, then it's going to have red algae. And this red algae, we have it here, is going to have phycoerythrin. And phycoerythrin is a pigment. So in absence of this phycoerythrin, this algae, red algae, actually look green because they have chloroplast. But in presence or in abundance of this pigment, if the algae are producing tons of this phycoerythrin, they are going to look red or even blackish. Have you eaten sushi? Okay, red algae, or the layer of sushi, the nori that we call, is red algae. Green algae don't produce this phycoerythrin, so they look green. The chloroplast is not masked in comparison with the red algae. In green algae, we're going to have chlorophytes and charophytes, and they look very green to me. And some of them, like in here, they can be very small. Some of them can be unicellular, and some of them can be multicellular. So this is the Chlamydomonas. It's not chlamydia. Remember, chlamydia is the bacteria. This is Chlamydomonas, is a algae, green algae. And then we have plants. And we're going to be covering plants extensively in later modules. And later we're going to have Uniconta, which has amoebozoans and opisthokonts. And opisthokonts actually include humans in Animalia, fungi, and other groups. But we're going to be covering that later in the second half of this semester. I have put this table for you to learn all of the different structures, the 4 supergroups in this new classification. These are the supergroups of eukarya, the domain eukarya, the major groups inside of this supergroup, and the key morphological characteristics that define each of these groups and specific examples. So if I can give you an advice on how to prepare for the exam and how to succeed, it will be to look at this table and remember all of these, plus the importance, the ecological importance. So these are the same tables, but just a section of it. I hope that you have enjoyed this class and you now understand why protists are an artificial group and the importance of primary and secondary endosymbiosis, how genetics has led to this reclassification of the protists and the reclassification of eukarya to form these 4 supergroups. Also, you learn that there are several parasites that affect humans, and also others that have a very important role in the ecosystem and that we depend a lot from them. Please complete the additional videos to have a better understanding of protists. Thanks.